into NCBI to, okay, that submits into NCBI and provides the bioproject accession with that submission. So for example, this one, sorry about the phone. So example, this one has these two biosamples, and this biosample has SRA data and an assembled genome. This one just has SRA data. So that's a basic bioproject. And we have the ability to cluster bioprojects together underneath an umbrella. And in fact, you can have multiple umbrellas. So you can have a relatively complex hierarchy if that is actually relevant and important for your particular project. Most of the things that we have in bioproject are just standalone single data level projects. Okay, so I know from the chat that Logan had forwarded to me that there was some discussion about like, how do we decide how to put them together? And there are quite a number of factors that that come into play. And so I've listed the ones that came to mind when I was putting this together. So, I mean, the first one, which actually relates to you guys, is that some organizing group or committee has said, okay, here's the structure, you know, submit inside this structure. Someone's already given you the rules. When you have to do it yourself, then these kinds of things come into play. So, for example, if it came from different PIs or different grants or different funding sources, then you can imagine you'd have separate ones for each of those. If you have different kinds of data, like maybe you want to put your transcriptome data all together over here in this bioproject, and your raw reads over here and your geodata over there or something. So that might be another way to do it. A lot of times they're grouped by citation. So if you know this data is going to be in paper 1, then maybe that's in this project, and you would have a different project for a different type of data. But that's really probably not relevant for this group, but that is an option. The volume of data can also make a difference too. So at one extreme, we have people who like they're interested in this particular fish, and so they're sequencing the genome and the transcriptome of that fish, and maybe they have some variation data or something. Well, that's not a good example, but anyway, they have a bunch of data just for this one fish, and so they put all that data into one project because there's no need to split it up. You just want all the data about that fish from these people. On the other hand, if it's a big project like this one sort of is, then well, this one definitely is, then you might want to have it goes back to the types of data. You might want to split your data apart into different projects. So that's another factor. And then finally, you might be thinking about how you want to group them for retrieval. So in the very first case, when we talked about the organizing group or the committee, those people might want it to be in a particular structure because they need to do metrics. They need to keep track, or analytics. They need to keep track of how much data they're getting and who is giving it to them or whatever. They may want it in a particular setup because that's how they're geared to check on the progress of the project or of the entire big project. I mean, another way would be, do you want to separate it by, in these big projects, do you want to separate it by who submitted it or by what kind of data does it contain? Or for example, what organism is it? Or maybe you have a bi-level thing and you combine these two. So anyway, there are lots of factors at play. And the bottom line is that we don't really have a preference. I mean, it's really up to the group who's doing the submission, how they want to arrange and organize their data and their bioprojects. So this is in my opinion. In my opinion, for these big projects, the best options seem to be that you organize by species and then by lab under that. And so that's what the FDA's genome tracker did. Or you could organize by species and then have all of the labs submit into the appropriate species. So this one has one less layer than this one. And then this was just my opinion on why, like just organizing by lab, and there are other things that you can do that are noisy, but when you have a lot of things involved, noise you don't really want to be noisy. It's better to be pretty clean. Okay, so this is just a little cartoon of that first option that I said. So in this case, we have this big umbrella, and then we have an umbrella for each species. And underneath each species, we have data projects. So the data projects are blue and the umbrellas are green. So we have data projects that each lab has its own data project. So lab 1 is submitting data for species 1 and for species 2. Lab 2 is submitting for species 1 and for, here's another umbrella that is just called metagenomic samples. So lab 2 is doing that, and lab 3 is only submitting here. But maybe in 6 months, lab 3 maybe will be submitting to all 3. Who knows? But so this is one way of that it can be set up, and in fact, that's what the genome tracker did. So this is from Ruth Timmy's paper. It's her figure. It's a very nice figure. So in this umbrella is actually the species 1 umbrella from my cartoon. So here we have the species of Salmonella enterica, and here's lab 1s and lab 2s and lab 3's data level project. And then their data is inside of each data level project. So these are the screenshots of what it looks like. Here's am I on the right one? No, here's what it looks like on our page. So here's this umbrella. It's underneath the Genome Tracker umbrella project, and it has 71 sub-projects of its own. So you can see that, here's Texas, here's Arizona, here's Emory. So that's one way to organize it, and it's a very nice way to organize it. Another useful way is the second option that I said. So here we have our umbrella, and underneath it, we just have data level projects for species 1 and species 2 and metagenomic in my example. And then all the labs would submit their species 1 data in here, and they would all submit species 2 here, and they would all submit metagenomic here. And so that's what the CDC, did I skip a little bit? Yeah, no, that's what the CDC HAI-seq project is like. So those are the screenshots, but again, here in their project, there's no umbrella. It's the top umbrella, but it contains 30 sub-projects. So for example, can you guys see this at all? Not that you really need to, but here's one for Bacillus species, Burkholdia cepacia outbreak, gram-negative bacteria. You get the idea. So CDC sets up, here are the projects that we are interested in. The people who are participating in this effort should be submitting their data to the right data level project. Okay. So for you guys, then, when you aren't involved in these big things and you have to make your own decisions about what it is you want to do, probably you won't be having this enormous structure. You'll just have a lot of standalone bioprojects that you have decided to put together because they have underlying, their organization makes sense in a particular fashion. So like if, Bill and Melinda Gates gave you $1 billion, you might put all of their data inside one bioproject. So these are just examples, but as I had said way back in the beginning, most of the time, most of our bioprojects aren't clustered together into this umbrella structure. And there's no reason, really no reason, to cluster them just for the sake of clustering. You would only do the clustering because they all belong to some overarching theme, and you want to be able to show that. And so that's the motivation for doing the umbrellas. Otherwise, you just have individual projects. And I personally like doing it by species and kind of data that's in there, but it's pretty much what you want to do. So then the next part that I was going to go into is what some NCBI pipelines do or use, how they use bioproject information. So one is the pathogen detection pipeline. And this actually goes to the paper that Ruth had pointed out in that chat, the FDA paper that she had pointed out in that chat. And so if you have something, a bioproject that you think belongs to, that you want the pathogen detection pipeline to incorporate into their system, then you send an email request to this address, and you give them the bioproject accession. And it can be, if there's a hierarchy in an umbrella structure, you can give them the umbrella, or if not, then you just give them the data level bioprojects. But when there's an umbrella, this pathogen system starts at the top and it works its way down, and it gets all the biosamples that belong to everybody underneath that umbrella, and it gets the data from SRA and it processes them in the pipeline. Another pipeline that uses stuff from bioprojects as a tool is the human scrubber for SRA data. So in this case, again, it's an email request. You send it to the SRA team at this address, but here it's the data, just the data level bioprojects. So this tool does not pay attention to hierarchies at all. So you need to give every bioproject accession for which you want to have this action done on your reads before they get, I'm not sure if it's accepted but basically before they get displayed. This tool, the human scrubber, is also on GitHub, so it's available. People can download it and run it themselves before they submit, which is very useful. Okay, switching gears a bit. So we have some tools that are useful for the bioproject submitters. So there's in the submission portal here, there's a tab called Manage Data here, and you can put in, it's tied to bioproject, SRA, and biosamples. And you can search with any of those 3 accessions, and you'll be brought to a page that has that information. So this is actually a fake thing in our developmental system. But anyway, what you can do in Manage Data is you can edit the title, edit the description, add grants and pubs. And so you don't have to send us an email to do simple things like that. You also can edit the SRA metadata in this system by mapping your way or clicking your way into the SRA data and making the appropriate changes. But in order to do that, you have to be the submitter of that bioproject, or you have to belong to the submission group under which that bioproject was submitted to us. And so this slide is just to go into that a little bit. So inside the submission portal, there's like the second page. When you start a new submission, the second page is you and your information. And there are 2 things I wanted to point out in here. One was that this one that's called Submitting Organization. This is really important because this is what appears in the bioproject as the submitter. So you would want to say like Wadsworth, State of New York Health Lab or whatever it's called. You wouldn't want to say Karen Clark here because it would show up as Karen Clark on the bioproject page, on the public bioproject page. So anyway, that's just to stress that. And that's only because I saw one bioproject where somebody didn't have the right information here. The other thing to point out is that when you do begin a new submission, you get this option once to make a submission group. And you can create a submission group, and when you do that, you're prompted to invite other people to join the group. And when other people join the group, everybody who's in that group can see all the submissions that belong to that group, and they can use the Manage Data page in order to edit the bioprojects that were submitted inside that group. So that's the very simple submission group that we have, that each submitter can make one of these and can choose to include a submission in that group or not. We also have a submission group that we can make for you. So we could make it at your request. And that one is, it's a similar sort of thing that the difference is that we create it and we invite you to join the group, and then you're the admin of the group. So then you invite everybody else that you want to join. They're pretty equivalent, but sometimes, we don't want, so sometimes you need to have 2 different groups because you're working with 2 different sets of colleagues, and so you want to have 2 groups. And So that's a way to do that, by creating your own and then asking us to make another one. Okay. What I wanted to point out on this slide was, so that all the genomes, all the eukaryotic and prokaryotic genomes go into NCBI's assembly resource, and this is its URL. And in the assembly resource, certain category, the genomes are all evaluated, and certain categories of certain situations, the genome is excluded from RefSeq. So it's basically tagged as, there's something a little bit off, or we just don't want to fill up RefSeq with this kind of data. So one example would be like the genome is really fragmented, way more fragmented than what we see for others of that species. So it's just like, okay, we're not going to, that one won't be promoted into the RefSeq world. But another one that actually pertains to this group is that we have a situation, I think now it's called derived from large multi-isolate project, or maybe just from large project. And what happens here is when there are 100 or more genomes of the same species in the same bioproject, then those genomes are not candidates for RefSeq. So they don't appear in RefSeq and they don't appear in the genomes resource. It's a really blunt tool, and its purpose is to not get flooded with the 250,000 pathogens that we have in GenBank, that the pathogen pipeline has submitted. But because it's so blunt, there are situations where clearly the person isn't doing a surveillance project. It's just that they have a lot of genomes of that species. And so if you think that your project is in that, if you think you have a project that's in that, has fallen into that bolus or into that bowl, then you can just send us an email and we can look and see, oh yeah, that's right, this is just an evolutionary study. It's not a surveillance project, and we can remove the flag, and then the genomes, appear, are normal candidates. They don't fall under this flag. Okay, and then my last slide was, I put this in red because in case you know anyone, we have an open developer position for Bioproject and we really need to fill it. So here's the ad for the developer. Okay, but when I was, so Logan had sent me some accessions of Bioprojects of people in this chat. And so I looked at a couple of them and noticed a few things. And I'll go through and show you what I noticed, but basically the take-home message of this slide is that we don't really have any monitoring of the bioprojects or their hierarchy. So things just get noticed by eye. So, if something is wrong and you can't fix it in the Manage Data tab yourself, then send us an email and we can probably fix it. And the final point here is that, however, moving an annotated genome from Bioproject 1 to Bioproject 2 is not a pleasant experience for us. So we don't want them to just drift around willy-nilly. So you really need to want to do it. Okay, so the examples, just to see what I saw. So yeah, so in this case, this is NCBI's COVID bioproject, and you can see that it has 54 data level projects, but then it has one umbrella project underneath it. So probably, and I checked with our team, and it's true. So probably these guys just made an umbrella because they thought, oh, I should make an umbrella, but no, we don't want that to happen. So we'll fix it for them, and we'll. So if we look what's underneath there, they have this project. So we'll just move this project to be directly underneath our COVID project, and we'll delete this particular. Whoops, sorry. We'll delete the umbrella. So that's easy enough to do. The other one is, this is the uppermost genome tracker umbrella. So it doesn't belong to anybody, and it has 19 sub-projects here. Where am I going with this one? Oh, I know what it was, yeah. So it has 16 umbrella projects and 3 data level projects. So this group made these projects and they linked them directly to the umbrella, which again is not the right thing to do. So we can fix it. I mean, we'll check with FDA to make sure that that's what they want us to do. But we can fix it so that this listeria goes underneath whichever of these is the listeria umbrella, and so that they go underneath the proper umbrella so that the structure is maintained. And that's it. That was it for me. So I just have the ad again for the developer position. But that's all I wanted to say. So I can hand it over to Nuala, and we can have questions now, and whatever you want, Logan. I'll stop sharing them. Oh wait, there's chats too.
Sure. Thank you so much for all of that information and for the great talk. Yeah, I don't know if Nuala, if you had anything else that you wanted to talk about, or if there were some kind of discussion topics that you wanted to maybe start off with. But otherwise, we can go into questions. So, Nuala.
Yeah, I mean, I think my understanding of this meeting is primarily about how to submit the bioproject. So maybe I'll just take a few minutes just to describe why I'm here and what my project is, and how it can be helpful to you guys, or you have more how you can be helpful to us. My name is Noelle OLeary, and I'm head of a small project within NCBI called NCBI Datasets. And it's a little bit different from other groups within NCBI, that we're not a curation group. We're actually a group that's redesigning all the web interfaces, command line interfaces, creating APIs for getting data. So we're trying to modernize the way in which NCBI presents itself, and the way in which people access data, web or programmatic. So I know some of you, I recognize some of these names. So we are the ones that put together the SARS-CoV-2 GenBank genomes, which I know a lot of people access directly from data sets. And the way in which data sets organizes data is that we like to take very simple entry terms that people know, the organism name, the bioproject accession, and give back people data, a package of data, a zip archive of data that includes, sequence, metadata, things that can be kind of complex. So I'm just to share my screen for just 1 second. Rather than slides, I thought I would just walk people through. This is a new project, it's only a few years old, but we already have a lot of things that are out in production. So this is our homepage. It's called NCBI Datasets. It is part of NCBI, so we're delivering NCBI data. But we're trying to, it's in beta, and we're trying to, work a lot with people that are retrieving data. So Karen is on the submission side, and we're on the data retrieval side. And so we have a web interface that's very focused around, searching for a species. You could just type it in, there's auto suggest. You get over to, these are new pages that we are developing that are organism centric. So these will serve as like a portal into data that's available for this particular organism. Karen mentioned genomes. We have a new genome table. So you come over here and you see all the genomes that are available for that particular, place in the taxonomic lineage. We have very basic filters for annotation, for reference. So we're trying to simplify the way in which people come to NCBI and follow those terms that they generally use to search, and make it easy for them to just come in and download. And you can do a table, you can do a data set, and by data set, I mean all the files that are associated with a particular assembly or genome. So if you're within this data sets environment, you also have access to our documentation. This is a description of our command line tool. So it's, again, this is a project that's in the beginning, and it's right now we have data for genomes, for genes, and for that specific SARS-2 data set. So we are very interested in submission, in that when people come to retrieve data, the better that submission is in terms of its metadata and how it's organized, the easier it is for people then come in and retrieve that data. So that's a really important thing to think about when you're thinking about submission and thinking about organizing your project. So we do have a way, say, for people to put in a bioproject ID, and they can get back a package of data that has all the genomes and metadata that are under that project ID, and that can be at the umbrella, the top of the umbrella, or underneath it. We have the whole hierarchy of bioprojects. So users can come and use this command line tool or the web interface and get all the genomes associated with that project. But for any given bioproject, there could be a diversity of data. Right now, we don't have the SRA data there. We don't have some of the transcriptomic data. We have the genomes and anything associated with the genomes. But we are very interested to know, like, what else would you like to retrieve? Like, what terms do you want to use? What data formats do you want? What tools do you need? So this project's under very active development, and we really want to hear from users as to what works best for them, and if this new resource is working for them. So I will stop sharing at that point, and that's my summary of data sets. So we'd love to hear from you guys.
Thank you. That was a great introduction to data sets as well. I know I had seen that webpage, but I hadn't really delved too deeply into it, and so it's nice to get a little bit of a kind of overview of what's going on there. And yeah, I think now I can probably open it up to questions if you guys are ready to take questions. And I figure I know that this call kind of came about because there were people who were kind of interested in getting some ideas about things that they were submitting, and so I'll open it up to the floor first, and if anybody has anything that any burning questions, go for it.
Sorry, I have a question. Could you go back to the data set slide?
Absolutely. To the homepage?
Yes. Do you have any, you have things that are organism based? Do you have anything for plasmids?
That's a great question. So the plasmid data is packaged in, if a plasmid is included in that genome assembly under GCA or GCF, it will be included in that package, but we don't have yet a way to search by plasmid. So on the existing NCBI website, there is a table, which maybe that's what you're familiar with that lets you look at like a table of plasmids. Is that the resource you're used to seeing?
Well, so like a use case. So recently for a project, we were looking at all of the KPC2 containing plasmids.
Okay.
And so I was just wondering if something like that would be part of the data sets.
I'm very interested in that. Yes. So we've had some requests for searching for a particular plasmid, or having a table of plasmids and being able to sort and filter through that. So yeah, if I'll put my email. I would love to hear sort of your workflow. What would be your entry term and what kind of data you would like to get back for that? That'd be really helpful.
So I think there was a question that came up in the chat, and then we'll go with maybe Sarah next. But so the question in the chat, I don't know if you can see it. I'll just read it out.
Okay.
How do you view metadata standardization? On the one hand, it seems critical to a consistent and complete database of metadata. On the other hand, it seems like it would be almost impossible task given the almost unlimited number of people submitting data to NCBI.
So I have one answer. Nuala may have more, but we created biosample in part to address that. So for the genomes and SRA data, people are required to give us sample metadata, but the values are in most cases are free text. So we have fixed fields, but free text values. So it's a struggle. Go ahead, Nuala.
I'm very interested in this topic because it's and I have listened to a lot of discussions about what to do about biosample because it's so chaotic, and maybe at this point is really hard to fix. Just one of the things that we did for this, so we have a new assembly page, is to add all that biosample information. Even if it's chaotic, add everything onto that assembly page. So you just have one place where you go and you can see all that biosample information. The sample information is also packaged. So when you download here, you can pick sequence files and annotation files, but you're always going to get a metadata report in every download. It's not a choice. It's you're getting that report, and in that report, it has all the biosample data for that assembly, and we deliver that data in a format called JSON lines, It's a very machine readable format. And we decided on this because the data, it's a way to get the data in a documented schema, even if it's a little bit chaotic. It has a schema. And we have a tool on our command line tool called data format. And that tool can be used to take that JSON lines file and convert it into a tabular file, which is a more friendly file, I know, for most people. So you know, we have to recognize that the data is getting very big, and that the details of that data, the metadata, is getting very complicated. So one of the things that's really important to us is that we at least document everything. So if you go back to data sets, we have documentation here, and you go into the reference section. And I will just point out that because this is under very active development, we're also looking at our documentation and making updates and making sure that it is working for people. Well, we have these report schemas. So for any given report, say genome assembly report, we're telling you what all the fields are. And then for that data format tool, and you know, if anyone is interested, we do workshops every once in a while, and you know, I know this is to go through sort of programmatic access takes a little bit longer, but we have all our fields documented, a description of what they are and how you can use that tool to take those specific fields you need and convert them into a tabular format. We're not really delivering tabular formats. You can from the web interface download a table, but we want the data to be structured and to make sense to people. So I hope that helps answer some of the questions.
Thank you. Sarah, do you have access to the mic? Can you?
Yes, yeah, I do. Can you hear me?
Mm-hmm.
Great. Okay. So I had a question, and I know this got brought up before, I think on StaPH-B Slack about the bioprojects for, like, I think that's great. And our plan is to, you know, submit everything based on a, you know, species level bioproject for Florida. But then we do get a number of, like, kind of one of our pet projects is looking at our CFIs, which are culture for identification, and many times using sequencing, and now even trying to use long read sequencing, we still don't have a definitive ID, or we might, you know, say, well, it's part of this genus, but we, you know, it's just many times it's something where the closest relative in NCBI, there's like one assembly or two assemblies for that particular potential species identification. So I guess my question is, what would be the cleanest way of organizing and submitting that in bioprojects? You know, because we could potentially have a bioproject that has, you know, 2 genomes for a really long time or something, you know, depending on what it is. Or do we just put that at the genus level bioproject or whatnot? Just looking for ideas to keep things in the cleanest, most organized way for people to find stuff.
Yeah, the genus idea seems like the best approach. Because as you said, you don't want to have bioproject proliferation. I mean, if they're clinical isolate, I mean, maybe there's some limit to how many you might expect, but it could explode. So having them at least be together in the same genus would give you some clustering, but also allow to avoid explosion. That would be my recommendation.
Okay, great. Oh, great. That was gonna be my approach, but because at first I thought about just putting all of our hard to identify stuff in one bioproject, but then I thought that would just get really cluttered really easily, I think. So I think just going the genus approach might be best. Thank you.
Yep. I mean, so for example, if they were coming from wastewater or something, you might want to just leave them in as a wastewater project, regardless of what it is, because the interesting fact is that they came from wastewater.
Right, yeah.
So you would have a lot of species under that. So that's sort of the tension, but yeah.
Okay, that makes sense. Yeah, these are clinical right now, but yeah, no, that does make sense about the wastewater too. Thank you.
There was another question in the chat
Right.
before we go on to the next one. What is the difference between data sets and e-utilities?
That's a great question. So we're not touching e-utilities. E-utilities is a command line API access to the what are the entree databases. So just to give an overview of NCBI structure. Let me trouble with it. Okay. So the old NCBI website it's a database of databases. So within each of these, there are these entree databases, and so the entree query system, that Boolean system that's very big and complex and allows you to get that very specific piece of data. That is an entry, and a programmatic interface to these entree databases. So datasets is looking at it differently. It's looking at this as one database and saying, what are the entry terms that people are using? So you don't need to know the structure of the entree databases to use datasets. You really just need to know your organism name. You need to know an accession. I'm trying to think what other terms we support. Those are the primary entry points or gene names. So we're using these more common terms, converting them into a list of accessions for data, and letting you access data across these databases. So that's I think the best way to look at data sets in terms of querying against it. The other thing is that utils is great for getting metadata and for getting small amounts of FASTA sequence. Data sets will let you download massive amounts of data. So you can get all salmonella genomes, and we have ways of dealing with these very large data sets, all 4.5 million SARS-CoV-2 genomes. So it's different in that way. That is letting you get a complex set of data. You can get multiple file types in one request. So, but it does not mean utils is going away. So it will still be there. And the entree databases will still remain for a while. So are there any questions about that? Okay.
So, is there somebody else? I think I heard somebody starting to unmute when I asked the question.
Actually, I have a quick question, if you don't mind.
Yeah,
Yeah.
go for it.
So related to the data sets, will there ever be a command line version? We do most of our analysis on a Linux machine that we don't use graphical interface for, but it might still be helpful to do a mass download using the data sets functionality instead of something like utilities.
Yes, we have our command line tools. So if you go to our documentation, there's a description of the data that we have right now available through our command line tool. We also have it in Conda. We have a GitHub site here. I can show you here, and that describes how you can go through Conda if that's where you want to use the tool. We just integrated into Galaxy as well, our genome service. So if you're using a Galaxy platform, you can get genomes through data sets within Galaxy. So yeah, this is out in production, and you can use it.
Cool, thank you.
Yeah, and please contact me if you have any questions. We're happy to help or meet with anybody.
I have a quick question about the bioproject stuff, and I was curious, is there like a visual browser that one might be able to use to kind of explore the umbrella projects and sub-projects?
No, unfortunately, we don't have that. It's just one by one, up and down through the, we don't have a cool graphic of, cool thing. We can put that on new list.
It is on our roadmap.
Yeah, exactly.
So bioprojects interface is also under my project. So again, yeah, if you have ideas and things you want to see, we are imagining that the web interfaces that Datasets is making could also, support this sort of nicer navigation through bioprojects.
I just kind of akin it to working with a relational database, or having some sort of map to kind of guide you across the system would just be really helpful.
Yeah.
Are there any other questions that people had? I guess in the meantime, if people are thinking of other questions, one thing that I did kind of want to ask about, and I think we kind of got to this a little bit before, but it sounds like, Ruth Timmy's group is kind of trying to establish some kind of guidelines surrounding, submission and use of bioprojects. but from your talk, it sounded a little bit more like you are staying a little bit more agnostic in your approaches. Are you guys looking at potentially like adopting any sort of like best practices or standards or like kind of, I guess, suggestions or guidelines or things like that? Or is it pretty much just kind of like a, as much as you can?
Wild Wild West. Yeah. We haven't really thought about going towards suggestions, but I think Ruth and I have very similar approaches. So I always like what she says. So she has very good ideas. So.
I know.
But yeah, it would be worthwhile to do something, have a little bit of more guidance at our side. So I think health documentation's in my purview, not Nuala's. So.
I wonder if you guys have had any interactions with like the phage working groups at all. I know that she was kind of working with them, or if that was something that was on your radar.
So that's not me personally, but yes, my coworkers. Yeah, but not me personally. Yeah.
Okay, thank you.
Yeah, I'm sure they'll feed back to us, so that'll be good.
Yeah, I mean, my only comment on that would be, I know the submission process can feel exhausting, and but for consortium, as much as you can enter data that has the same field name and that the entry is structured the same way, the better it is for people when they're coming in to get that data. And we allow, in NCBI, we allow the user a lot of freedom. And that's something I know that we're thinking about restricting in some ways. And we have templates like for the SARS-2 genomes, but we don't, those templates are not too restrictive on the user, and that ends up creating some of the chaos.
Yeah, we're very much permissive.
Yes,
yeah.
A little too permissive. So it is a discussion within the institution about how to handle it.
Yeah, I can see
So,
either way.
Mm-hmm.
So like with the pathogen, with the genome tracker, the direction is to use the particular biosample packages, right? Which is one way to help drive towards commonality, but it's the same, it's what Nuala was saying, yeah.
Biosample data is very important, and it's very important that it be consistent as you can, at least within a consortium. Makes a big difference.
Yeah, because the data it's not worthless, but it's worth so much less without the metadata. So
yeah.
Thank you. All right. Is there anything else that anybody has questions on? I know that it looks like Nuala put your information in the chat, and I can send that out to everyone on the call afterwards too, if people are too shy to ask questions on the call, or if you think of anything that kind of comes up afterwards.
No, you can just Google NCBI datasets, you'll find our page.
Mm-hmm.
That's our goal, Googleable.
All right. Well,
I can put mine there, but I'll put 2 there because it's better to write to an alias in our case, because but anyway, yeah.
Okay. Yeah, and I will go ahead and copy those down too, so that I can send those out afterwards with the call information as well. All right.
Okay.
Well, I can hang out on the call for a few minutes afterwards in case there's any other kind of burning last minute questions. But if not, then I think that we can go ahead and give people back maybe 5 minutes of their day. Thank you so much to our speakers for being on today. I really appreciate you guys answering questions and giving us this great overview. It was really helpful. And I think that a lot of people get a lot of utility out of this for our groups. So, and we're excited to, kind of explore more around with data sets. And I look
If you're
forward to-
interested in us doing a longer like hands-on tutorial that we have, we've built Jupyter notebooks and other tools that require more a longer sort of workshop, but we're happy to arrange that if that's helpful to people.
Okay, yeah, we can potentially get in contact with you, and maybe specific groups might have more utility for those kind of trainings where they would have more kind of use cases that you guys would be able to help them out through.
Yeah, particularly if people are interested in using the command line and some aspects of that would be good to do in a tutorial.
All right.
All right.
Well, thank you so much.
Okay.
Happy Friday, everyone.
Thanks, everyone. Bye.
Thanks very much. Bye-bye.